Okay, so we continue with the first pathway, which is the Emp pathway. So basically, this is the most familiar or the most common glycolysis pathway. Okay, used by brewers, bread makers, and cheesehead. This pathway is operated by Saccharomyces to produce ethanol and CO2. Basically, the alcoholic fermentation. This pathway is also used by the homolytic bacteria, means that this bacteria can only produce lactic acid. Okay? And some bacteria can undergo heterolactic acid fermentation, means that it can produce ethanol and CO2 on top of the lactic acid, okay, as in the homolytic acid bacteria. And the formation of this component usually by using other kind of pathway, not Emp pathway anymore, which is the Phosphor Ketosis pathway. Some other, and of course, some bacteria can also produce other acids as well, and fatty acids, alcohol, and gases. This one usually we call them as the mixed acid fermentative bacteria. Some end product of this fermentation are essential components in food and beverages. For example, in fuel and industrial solvent industry. Okay, so as you can see from here, these are the two examples of the pathway that we're going to take a look at. The first one is Emp pathway, very familiar, very common. Glucose is going to be broken down into basically glyceraldehyde 3-phosphate. Okay, this is what we call as branching point, and eventually both of them will produce the pyruvate or pyruvate acid. This pyruvate can be used to produce other components such as lactic acid in the microorganism. Okay. The second one, as you can see here, is the comparison between the Emp pathway and also the Eddy pathway, the second one. As you can see here, they both can produce eventually the pyruvate. They can produce pyruvate or they can produce the byproduct after that, such as ethanol and lactic acid. Only, as you can see from the right one, is the components that are different from one fermentation to the other. Okay, as you can see from here, glucose 6-phosphate produces 6-phosphate, right, for the Emp pathway. But for the Eddy pathway, glucose 6-phosphate will produce different product using different enzyme. That's why you can see different number here. This enzyme is number 2, but this enzyme is number 5. Okay, so basically all the fermentation pathway, they all will produce similar end product, which is pyruvate. Only the intermediates is going to be different. Okay, this is the explanation of the formation of the pyruvate from the common glycolysis. I think I don't have to elaborate more on this because you learned about this during your basic biochemistry lesson. I just want to say here that the oxidation of glucose to lactate yields a total of 56 calories per mole of glucose. Since the cells harvest only 2 ATP, because the other 2 is used for the investment stage of the glycolysis, as useful energy, the efficiency of the lactate fermentation is only about 29%. And alcohol fermentation have a similar efficiency because they also produce 2 ATP. So I just want to show here that anaerobic respiration in microorganism is much less efficient than aerobic respiration that happens in the human, okay, or in certain microorganisms as well. Okay, so this is the example of the processes. The first one is glucose will eventually produce pyruvate acid, and lastly it will produce 2-lactic acid in a homolytic fermentation. Okay, in other microorganisms, for example Saccharomyces or yeast, glucose will produce pyruvate acids eventually using the same processes, but instead of producing fatty acid, it will produce alcohol and also CO2. This is one of the main example is the production of bread. Okay? The bread is being fermented by yeast, right, Saccharomyces, and the bread will rise because of the presence of CO2. So the bread that we consume actually contains ethanol, but this ethanol is actually being burned off during the baking process. So we as a Muslim, we can still consume this bread because most of the ethanol is not present anymore in the bread. Okay, so this is the example of the processes. For example, pyruvate will be turned into lactic acid, or in yeast, pyruvate first it will turn into acetaldehyde before it can be converted into ethanol. Okay, mainly used in bread and also in the brewery. Okay, alcoholic fermentation to produce alcoholic drink. Okay, this is the example of the reaction that is happening. The first one is in homolytic bacteria. Okay, just produce lactate, and the second one is heterolactic bacteria that can produce ethanol on top of lactic acid. Okay, besides lactic acid, EMP fermentation in bacteria can lead to a wide array of end products depending on the pathway taken in reductive steps after the formation of pyruvic acid. Okay, so the formation of other product will happen after the formation of the final product in glycolysis. Okay, so it is usually known as mixed acid fermentation. Okay, the other two are homo and heterolactic. Okay, so remember, pyruvate is the most important precursor for all the fermentation processes. As you can see from this graph, for example, the pyruvic acid, they can produce, major one is lactic acid. Okay, and also they can produce some kind of alcohol, for example, butanediol or butanol. Okay, they can produce other kind of acid, for example, butyric acid. Okay, and propionic acid, they can also produce isopropanol, one kind of the alcohol. Okay, so in homolytic fermentation, lactic acid is the sole end product. As I told you before, the bacteria are used to ferment milk and milk products in the manufacturing of yogurt, buttermilk, sour cream, cottage cheese, and so on. Okay, basically anything that utilize the lactic acid. Okay, in this case, usually the milk product or dairy product. Okay, mixed acid fermentation, mainly the pathway of enterobacteria. Enterobacteria is usually the gram-negative bacteria. Okay, one of the main or the common example is E. coli. So the end product can be a mixture of lactic acid, acetic acid, many kind of acids, and also sometimes it can even produce ethanol or alcohol, with the possibility of the gas formation, for example CO2 and H2. If the bacterium possesses the enzyme formate dehydrogenase, which cleave formate to the gases. So basically, enterobacteria is one of the most complex one or the most diverse one that can produce many kind of product compared to the other two. Here, one of the example is propionic acid fermentation. This is an unusual fermentation carried out by propionic acid bacteria. Although sugars can be fermented straight through to propionic acid bacteria will ferment lactate, the end product of lactic acid fermentation, to acetic acid, CO2 and propionic acid. Okay, because this is quite rare, only certain bacteria can undergo this process. The taste and the flavor of the food that you produce from this kind of fermentation is also unique. Okay, even the processes are very unique. The formation of propionic is a complex and indirect process involving 5 or 6 reactions after the formation of pyruvate. Overall, 3 moles of lactate are converted to 2 moles of propionic plus 1 mole of acetate plus 1 mole of CO2 and 1 mole of ATP is squeezed out in the process. The propionic acid bacteria are used in the manufacturing of Swiss cheese, which is distinguished by the distinct flavor of propionic and acetate and holes that caused by the entrapment of the CO2, because it also produces CO2 as well. So this one is the heterolactic or phosphor ketosis pathway, okay, in certain bacteria. As you can see from this one, they produce different kind of intermediates. Okay, and this process, the cleaving into 3 molecules of carbon from glucose, undergo a specific process called phosphor ketosis compared to the normal ones. It's a different name, and eventually they will also still produce pyruvate, okay, and produce lactic acid. But if the other pathway or the other set of reaction, they produce ethanol without the formation of the pyruvate, but they produce the acetyl-CoA first, okay, and produce acetaldehyde and eventually ethanol. So the heterolactic species of bacteria are occasionally used in the fermentation industry. For example, kefir, a type of fermented milk to yogurt, is produced by using a heterolactic lactobacillus species. Interestingly, I ate this kefir once this year. I bought it in Aeon, and it says it's halal. But based on our lecture here, we know that some of the bacteria can produce the ethanol as well. So probably they can evaporate or remove the ethanol somehow so that Muslim can consume it. The taste is very distinct compared to yogurt. Tastes very gassy, and also the taste of acid is stronger than the normal yogurt drink. Okay, you can try and go to the Aeon and buy this kefir. Quite expensive because this one is quite unique. And it is produced by FGV, yeah, Felda. Sauerkraut and kimchi. Sauerkraut is kind of fermented vegetable similar to kimchi, but usually it is used in European countries. Use leuconostatic, a heterolactic bacterium, to complete the fermentation. So even kimchi contains the bacteria, so I'm not sure if it contains the alcohol or not. Probably in a small amount that we Muslim can eat it. Okay, next we take a look at the Eddy pathway that I told you before. It's just similar to the Emp pathway, just the intermediate is different. The end product is usually the same. Only a few bacteria, most notably Zymomonas, employ this Eddy pathway as a strictly fermentative way of life. Means that it's not going to undergo the aerobic respiration at all. Although they have the oxygen, they still prefer the fermentative way of producing the energy. Means that they still prefer to not entering the TCA cycle and etc. However, many bacteria, especially those group around the Pseudomonas, use pathway as a way to degrade carbohydrate for respiratory metabolism. The Eddy pathway yields 2-pyruvic acid from glucose, same as Emp pathway. Okay, it means that the glucose will be broken into 2 molecules of 3 carbon. But like the Phosphor Ketosis pathway, oxidation occurs before the cleavage, and the net energy is 1 mole of ATP per mole of the glucose utilized. Alright, so total is 2 mole because it's broken down into 2 pathways. In Eddy pathway, glucose phosphate is oxidized to different kind of product, KDPG. Okay, as you can see from here, KDPG should be around here. Okay, this one, right? KDPG. Okay, and broken into pyruvic acid. Okay, and eventually we produce ethanol and ethanol as well. So they will produce 2 molecules of ethanol. The latter is oxidized to pyruvate by pyruvic acid from either branch of the pathway is reduced to ethanol, in the same manner as yeast by the yeast-like bacterium Zymomonas. So usually this bacteria is used in the alcoholic fermentation. For example, in the alcoholic beverages formation or production. Okay, the reaction is glucose will produce 2 ethanol plus 2 CO2. Oh, actually just produce 1 ATP instead of 1. Okay, so probably the ATP is produced here instead of here. So only this pathway produce ATP as compared to the Emp pathway, which will produce ATP at both sides of the reaction. Okay, this is the example of the usage of Zymomonas. It's being used in, I think, Mexico. Okay, this one to produce the alcoholic drink. Yeah, because this Zymomonas is actually present naturally on this plant, and that's why it's being used in the production of this special alcoholic drink. And of course, because we use different kind of microorganism during this alcoholic drink, the taste will be different. For example, the taste of the alcohol drink in Mexico will be different from the one that is being produced using the yeast. Okay? As it is said here, Zymomonas has potential advantages over yeast for industrial production of alcohol, but industry is yet to do what it can do. Means that they are doing this one traditionally, so they don't even know why the taste or the flavor can be different, because Zymomonas can produce special flavor. And also Zymomonas was also shown to be able to use different kind of substrate compared to the yeast, the normal yeast. See, I heard you can use, for example, different kind of sugar. I forgot the name of sugar, xylose. Okay, which cannot be used by the normal yeast. Okay, so it is quite special. Okay, thank you. We will meet in the other lecture.